Hello everyone, welcome to another lecture on carbohydrate metabolism. Here today we are going to see about glycolysis. In glycolysis, we will see how glucose is converted into pyruvate through a series of reactions which are catalyzed by enzymes. In this process, we have two ATP molecules produced per glucose and it also occurs in the cytoplasm and does not require oxygen. Yes, you are right, you heard it right. It is an anaerobic reaction. Let's see about the glycolysis. Here, the first substrate is the glucose. So what I will advise you is, I will advise you to learn all the steps wholeheartedly, because in exams, orals and viva, the examiners will ask you, or else even in exams, they will ask you about each and every step or the enzymes, or they will trick you with the questions which will require you a full knowledge of this glycolysis steps. I will tell you how to remember each and every step. Okay, let's get started. So this glucose gets trapped into the cell by the addition of phosphate. So once phosphate is added to the glucose, it forms glucose 6 phosphate. So in biochemistry, for a phosphate to get added or removed, that enzyme will have the suffix kinase. So for the glucose to get converted to glucose 6 phosphate, we have the enzyme hexokinase or glucokinase. And for the glucose 6 phosphate to get converted to fructose 6 phosphate, so glucose and the fructose are isomers, isn't it? So the enzyme is known as phosphoglucoisomerase. Fructose 6 phosphate forms fructose 1,6 bisphosphate. What happens here? If you notice, here there is only one phosphate in the sixth group. Here there is addition of another phosphate in the first group. So here since the phosphate is added, the enzyme will have the suffix kinase. So it is known as phosphofructokinase 1, that is PFK1. You have to remember that it is of 1 because we have another PFK2 which also plays an important role in glycolysis. Now this fructose 1,6 bisphosphate is broken down into two compounds by the enzyme aldolase. It forms dihydroglyceraldehyde 3 phosphate as well as dihydroxyacetone phosphate. They both are interchangeable by these isomers. So triose phosphate isomerase converts glyceraldehyde 3 phosphate to DHAP and DHAP to glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate by the enzyme glyceraldehyde 3 phosphate dehydrogenase will be converted to 1,3 bisphosphoglycerate. This bisphosphoglycerate, see here 1,3 bisphospho. There are two phosphates in first and the third position, isn't it? So the first position phosphate is removed. That is done by the enzyme phosphoglycerate kinase. So how do you remember this? See, this is phosphoglycerate kinase removes this ATP and therefore 3-phosphoglycerate is formed. Once this is formed, the next product is 2-phosphoglycerate. What happens here? The phosphate in the third position is moved to the second position. This is by the enzyme phosphoglycerate mutase. When there is a change of position, the enzyme that is involved is mutase. This 2-phosphoglycerate with this enzyme enolase will form phosphenol pyruvate. And finally, we have our final product pyruvate by the enzyme pyruvate kinase. In this slide, we are going to see about the difference between hexokinase and glucokinase. Hexokinase and glucokinase is the enzyme which converts glucose to glucose 6 phosphate. So, if glucose is going to get converted to glucose 6 phosphate, why should we require two enzymes? We have two enzymes which cause this conversion. This is because hexokinase is the one which is present in all the cells, but glucokinase is the one which is present in liver cell. The second thing which you should know that the hexokinase has a high affinity to glucose. Why? Because once the glucose is presented to the bloodstream, then what should ideally happen? All the cells should take up the necessary glucose from the bloodstream into the cell, isn't it? So it should have a high affinity to the glucose. High affinity, anything which has a high affinity is inversely proportional to Km. That means it has a low Km. Similarly, when the glucose is presented to the cell, the liver should not be in a rush to take up the glucose into the liver cells. Then all the other cells will starve, isn't it? Logically speaking, this is how the concept is, right? So the glucokinase is the enzyme which is present in liver cell and that has a low affinity affinity to glucose because it waits for all the other cells to take glucose into the cell and convert it to glucose 6-phosphate. What is the third important point? Third important point is low Vmax. What is a Vmax? Vmax is the maximum capacity that this enzyme can take up. Okay, so this hexokinase which is present in all the cells should have a low Vmax because it should take up only the amount of glucose which is needed for the cell. It should not take a maximum capacity. It should not take the maximum amount of glucose into one particular cell, isn't it? So even though it has high affinity, it will have a low Vmax. But on the other hand, if you see the glucokinase which is present in the liver cell, it will have a low affinity because it will wait for the cell or the other cells to take up glucose, and it will have a high Vmax. Why? It will have a high maximum capacity to take up the remaining glucose because we don't want the glucose to stay in the blood, isn't it? Then we develop the condition called diabetes. So the glucokinase which is present in the liver helps to clear the glucose by having a high Vmax. The other important factor is hexokinase is not upregulated by insulin, but glucokinase will be increased with insulin. So even though the glucokinase has a high Vmax, okay, but still if there is if the glucose in the blood is high, then what happens? Then if there is insulin, then the glucokinase is upregulated and it will take up the glucose into the liver cell. How is this knowledge asked in the question? It is tested in exams by this type of question. 17-year-old female with a frequent thirst and urination. So they show that she is having an uncontrolled diabetic plus markedly elevated serum glucose and ketones. So that means what? Ketones are there. So diabetic ketoacidosis, elevated glucose, it is an uncontrolled, and a serum pH of 7.1. It also implies that they are having an acidosis. Following administration of insulin, activity of after administration of insulin, activity of which enzyme is increased. Once insulin is administered, glucokinase is increased, and that will cause a conversion of glucose to glucose 6-phosphate. We have already seen about glycolysis. Now, in glycolysis, there are certain enzymes which are irreversible. These irreversible enzymes are the enzymes which will regulate glycolysis itself, isn't it? Otherwise, this reaction will be going forward and backward and forward and backward. Only if there are irreversible enzymes, then they will catalyze a forward reaction alone, isn't it? So, and they are also essential for glycolysis. Any irreversible enzyme, most of the time, is called as the rate limiting enzymes. So what are those enzymes? Is irreversible enzymes in glycolysis. An ideal way to memorize this is first, third, and the ninth enzymes are irreversible. First is the hexokinase or glucokinase. Third is the phosphofructokinase 1, and the ninth is the pyruvate kinase. We had already seen that there are three irreversible enzymes in glycolysis, isn't it? So the pyruvate kinase is that ninth enzyme, and once the pyruvate kinase activity rises, that is the key step in glycolysis. So what is the metabolite that the pyruvate kinase acts? Pyruvate kinase will act on phosphenol pyruvate to pyruvate. Now here you should know that how this concept is asked in questions. 23-year-old male consumes energy drink. Which of the following irreversible enzymes is most likely to demonstrate increased activity? When there is an energy drink which will mostly contain lot of sugar and excess of glucose, then the pyruvate kinase is that irreversible enzyme which is likely to demonstrate increased activity. This is how they will ask you in USMLE to test your applied knowledge. What are the molecules which stimulate glycolysis? By glycolysis, we have energy being produced. So low energy states will stimulate glycolysis, such as AMP, adenosine monophosphate, ADP. It will also indicate that ATP is depleted, isn't it? And fructose 2,6-bisphosphate will also stimulate glycolysis. Let's see about it in detail in a short while. Let's see how fructose 2,6-bisphosphate functions to stimulate glycolysis. So in glycolysis, glucose is getting converted to glucose 6-phosphate. Glucose 6-phosphate gets converted to fructose 6-phosphate, and with the help of phosphofructokinase 1, fructose 1,6-bisphosphate is formed, isn't it? This is what our knowledge from the previous slides. Now when there is excess of glucose which is present in the blood, then insulin is released. This insulin will trigger the formation of fructose 2,6-bisphosphate. So from how is fructose 2,6-bisphosphate formed? When it's excess of glucose, and when it forms fructose 6-phosphate, and it is waiting for phosphofructokinase 1 to convert it to fructose 1,6-bisphosphate. In the meantime, since there is excess of fructose 6-phosphate, some of it is also converted to fructose 2,6-bisphosphate by insulin. So the role of 2,6-bisphosphate is to activate phosphofructokinase 1 so that it will accelerate the glycolysis process, and whatever glucose is present in the blood is cleared off with this glycolysis. What is that which will inhibit glycolysis? Glycolysis is a pathway to produce energy. So high energy levels will stop the glycolysis. What do we mean by high energy levels? High levels of citrate will indicate abundant energy. How? This is because the end product of glycolysis is pyruvate, which forms acetyl-CoA. Acetyl-CoA will combine with oxaloacetate to form a citrate, which will enter the citric acid cycle and produce energy. So now when there is a high amount of citrate, it means that there is already high pyruvate as well as which has already formed acetyl-CoA. So henceforth high citrate will indicate abundant energy, and therefore high citrate will stop glycolysis. How does it stop glycolysis? The excess of citrate will move to the cytoplasm where glycolysis is happening, and it will inhibit one of the rate limiting step of glycolysis, which is the phosphofructokinase 1. Hence, citrate acts as a feedback inhibitor, preventing the unnecessary breakdown of glucose when the cell has enough energy. The second molecule that will cause an inhibition of glycolysis is alanine. How? Alanine is formed by transamination from pyruvate. By glycolysis, the glucose is broken down into many intermediate steps and finally forms a pyruvate. So once pyruvate is formed, that undergoes transamination to form alanine. Pyruvate forms many products like pyruvate forms acetyl-CoA, pyruvate forms lactate, and pyruvate also forms alanine by transamination. High alanine levels indicates a sufficient energy stores, isn't it? So when the pyruvate is high, only when pyruvate is high, alanine can be high. When alanine is high, it indicates that the product is in high amount and is excess. So therefore we don't want this pathway to happen. So this alanine, how does it inhibit glycolysis? It inhibits glycolysis by preventing one of its rate limiting step, such as pyruvate kinase inhibition, and slows down the glycolysis. We have come to the end of our glycolysis topic discussion, and we will finish it off with a question which is asked in USMLE. A group of researchers are investigating a protein in the RBCs that shifts the hemoglobin oxygen dissociation curve to the right. Okay, it is concluded that this molecule can be synthesized from a glycolytic intermediate via an RBC enzyme called bisphosphoglycerate mutase. So there is a glycolytic intermediate from which that molecule is synthesized. So which of the following glycolytic substrates is most likely the precursor to the RBC protein? This is what the question is. So the enzyme is bisphosphoglycerate mutase. So this bisphosphoglycerate mutase will convert 1,3 bisphosphoglycerate to 2,3 bisphosphoglycerate, which will again can get converted to 3-phosphoglycerate. We know that 2,3 bisphosphoglycerate, which is abbreviated as 2,3-BPG, is that protein which will shift the oxygen hemoglobin dissociation curve to the right, isn't it? So that's what they are asking, and they are telling you that the enzyme, RBC enzyme, is bisphosphoglycerate mutase. And now what do they want to know? They know the 2,3 bisphosphoglycerate is a product. bisphosphoglycerate mutase is the enzyme which forms this product. Now they are asking which of the following glycolytic substrates is most likely the precursor. So they want to know about this. So this is the most likely precursor of this enzyme. 1,3 bisphosphoglycerate is the answer. Okay, so the RBC protein 2,3-BPG is some 1 is the answer, and some 1,3-BPG from glycolysis is converted to 2,3-BPG inside the RBCs via this enzyme bisphosphoglycerate mutase. Mutase is what? Mutase is whenever there is a change in position, like when 1, 3, the atoms which are present on the 1 and 3, when it gets changed to 2, 3, that is usually done by the enzyme with the suffix mutase, isn't it? So in that way also you can remember it, like this must be the precursor protein, since this is the enzyme. That is a shortcut to answer questions like this. Thank you for being with me all this while. Now we have come to the end of the glycolysis chapter. I have an announcement for you. I am starting my mentoring lessons to medical graduates as well as USMLE aspirants. If you like my videos and if you want to have one-on-one lecture with me, or if you want to get mentored by me, we can discuss further and proceed it. You can go ahead, send a message in the comments and fill my Google form. Looking forward to see you succeed. Have a good day.